University at Buffalo scientists have discovered a convergent mechanism that may be responsible for how two top-ranked genetic risk factors for autism spectrum disorder, intellectual disability, ASD, ID, lead to these neurodevelopmental disorders. While ASD is distinct from ID, a significant proportion, approximately 31%, of people with ASD also exhibit ID. Neither condition is well understood at the molecular level. Given the vast number of genes known to be involved in ASD, ID and the many potential mechanisms contributing to the disorders, it is exciting to find a shared process between two different genes at the molecular level that could be underlying the behavioral changes, said Megan Conrow-Graham, Ph.D. Published today in the journal Brain, the paper focuses on ADNP and POGZ, the two top-ranked risk factor genes for ASD, ID. The research demonstrates that mutations in these genes result in abnormal activation and overexpression of immune response genes and genes for a type of immune cell in the brain called microglia. Our finding opens the possibility of targeting microglia and immune genes for treating ASD, ID, but much remains to be studied, given the heterogeneity and complexity of these brain disorders, said Zhen Yan, Ph.D., senior author and SUNY Distinguished Professor in the Department of Physiology and Bi. The research involved studies on postmortem brain tissue from humans with ASD, ID, as well as studies on mice in which ADNP and POGZ were silenced through viral delivery of small interference RNA. Weakening a repressive function. Under normal conditions, cells in the central nervous system should not express large quantities of genes that activate the immune system, said Conrow-Graham. When that repression is weakened, these immune and inflammatory genes are then able to be expressed in large quantities. The upregulated genes in the mouse prefrontal cortex caused by the deficiencies in ADNP or POGZ activated the pro-inflammatory response. This is consistent with what we see in upregulated genes in the prefrontal cortex of humans with ASD, ID, said Conrow-Graham. The prefrontal cortex is the part of the brain responsible for executive function, such as cognition and emotional control. The mutated genes also activate the glial cells in the brain called microglia, which serve as support cells for neurons and have an immune function in the brain. They comprise 10 to 15% of all brain cells. The researchers pointed out that the clinical presentation of both ASD and ID is incredibly varied. Significant variation also likely is present in the kinds of mechanisms responsible for the symptoms of ASD and/or ID. When I began my PhD, I had completed 2 years of MD training, so I was familiar with the basics of physiology, anatomy and pathology. Because of this, I was able to bring a broader perspective to my neuroscience research, identifying how the immune system might be playing a role. Prior to this, our lab had not really investigated immunology-related pathways, so having that background insight was really beneficial. She added that she learned so much from all of her colleagues in Yan's lab, including faculty members, lab technicians and other students. I learned so many technical skills that I had never used before joining the lab, thanks to the dedication of lab coworkers for my training, she said. Her experience at the lab bench working on the basic science underlying neuropsychiatric disorders will definitely influence her work as a clinician. I plan to pursue a career as a child and adolescent psychiatrist, so I may be able to work directly with this patient population, she said. We're learning now that better care may be able to be provided by taking a personalized medicine approach, taking into account genetics, psychosocial factors and others. Being able to take a very deep dive into the field of psychiatric genetics was a privilege that I hope will help me to provide the best care for patients. The research was funded by the Nancy Lurie Marks Family Foundation and by a National Institutes of Health Ruth L. Kirschstein Individual Predoc. In addition to Conrow-Graham and Yan, co-authors are Jamal B. Williams, Ph.D., former graduate student, Jennifer Martin, Ph.D., former postdoctoral fellow, Ping Zhang, Ph.D., senior research scientist, Ching Cao, Ph.D., postdoctoral fellow, and Benjamin Rain, Ph.D., former graduate student. All are current or former members of Yan's lab. Materials provided by University at Buffalo. Original written by Ellen Goldbaum. Note: content may be edited for style and length. Brain, 2022. doi. 10.1093. Brain, AWAC 152 University at Buffalo. https://ift. tt. 6zka8 high. Access July 1, 2022. Thanks for watching. Please subscribe my channel for more videos.